Hello, everybody. Welcome. We are going to have a quick lecture about an introduction to the nervous system. So, what's going on with our brain? What's going on with our nerves? How do we move around? How do we do everything we do, really? So, first off, just to get us thinking about the right kind of stuff, what's going on in the nervous system of this cat? To see a string, to chase it, and attack it repeatedly. So how can me, you, every living thing that has a nervous system, how can it see things, respond to those things, tell itself to attack those things maybe? On your guided note sheet, or if you're taking blank Avid notes, this is your time to fill in the essential question, which is, how is our nervous system organized and how does it communicate movement, maintain homeostasis, and keep us functioning? And as always, we are going to talk about homeostasis as in every unit. So hopefully by the end of this lecture, you feel confident answering this question and specifically bringing it back to homeostasis, or that balance in our system. All right, what we are going to hear about today, we are going to split this into 4 sections, really. We're going to talk about organization, so how, what makes up our nervous system, how does it work together. We're going to talk about the anatomy of nerves, so how are our nerves built, what's happening at that cellular level, and we're also going to talk about how do our nerves send signals, how do they communicate, and finally, we're going to talk about how different chemicals can interact with our nerves. This can be in a variety of ways. Specifically, we're going to look at the impact of drugs on our nervous system, drugs that are legal, illegal, and maybe some that we produce ourselves as well. All right, the first one we're going to talk about is organization. So we're going to look at how our nervous system is made up, how do the things that are part of it, how do they work together, and keep us going each and every day. All right, just for this first part, we're going to think of all parts of our nervous system existing in really 3 steps. To do this, I like to imagine a stoplight. Say you're driving to school, you see a stoplight. That first time you see it, that's your sensory input, right? There's something in the world around you. Your senses are sending that information to your brain. Once we get to the brain, right, that's where these senses send information to integrate it there and process it there. So after you see that stoplight, you're not making a decision, okay, do I need to press the gas? Do I need to press the brake? And then the third thing is that motor output. And when I hear the word motor, I think of movement, right? So motor equals movement. And that's like our reaction to something. You're going to put your foot on the gas pedal or the brake pedal, depending on what light you see. So every part of our nervous system can really be broken down into these 3 steps. We are also going to look a little bit at nervous system tissues. All parts of our nervous system are made up of bundles of neurons, and that's the word that we are going to use when we are talking about nerve cells. So neurons is like that new word for us. It means nerve cells. These nerve cells conduct signals, or there's like impulses, we call them in this unit. And that's really how information is passed through each nerve cell. So from cell to cell. Those impulses help send information along. We also are going to talk a little bit about neurotransmitters, which are just chemicals that help pass information in these cells. And we're also going to talk about the black sheep of the nervous system, the neuroglia, which are important cells, but not quite the nerve cell. But they are going to provide protection, support, insulation, and nutrients for our neurons. So they're like working for our nerve cells. As we talk about the nervous system, we will also hear a lot about two different types of the nervous system, or like two groups of our nervous system. We are going to separate that into the central nervous system, which if you are looking at this first one, if you think about the word central, right, that means like right in the middle. So when we talk about the central nervous system, we're talking about the brain in the middle of our head. We're talking about the spinal cord, which is in the middle of our body. This is where things are sent to be processed. So if you've ever touched something hot and you reflectively like get away from that right away, those signals were sent to either our brain or our spinal cord to say, yo, get away, that's too hot. The other group that we're going to talk about in the nervous system is our peripheral nervous system. So this is everything else. This is all like the nerves that are in our face. It's the nerves that branch out from our spinal cord into our arms, into our legs, into everywhere that is on our body. These nerves, or this peripheral whole nervous system, is going to connect that central nervous system to other parts of the body. So that's what's making it possible for us to think something and be able to do it, even if it's far away from the brain. And we can further classify or organize our peripheral nervous system in a few more ways than we can with our central nervous system. So we first are going to separate it into two different kinds of divisions. One we're talking about is the sensory division, and the other one we're talking about is our motor division. If we say the word senses or sensory, we're talking about information in the outside world or even our internal world. The sensory division is going to bring information into the central nervous system. So this is going to be changes that are inside and outside the body. Part of this is monitoring our external environment for things like, hey, if there's a change in light outside, or if there's something we see, right? Light makes that possible. If we hear something different, those senses pick that up, and this sensory information is brought to the central nervous system. It's also about monitoring our body's internal environment. So if our body temperature drops too low, this is what's going to send signals to the brain that says we need to do something to bring that temperature back up. It's also going to monitor our oxygen levels. So if we are not bringing enough oxygen to our cells, what can we do in our body to increase that? The second part is the motor division, right? So motor, I think, again, movement. So motor is move. And this division conducts impulses from our central nervous system to responsive structures in our body called effectors. So we're going to see things like this in our muscles, right? Those responsive structures that can help us move, as well as in our glands. So some things that just are going to release maybe hormones in our body. These are both voluntary and involuntary motor functions. So this is moving voluntarily and moving involuntarily. So if we talk about voluntarily, we are talking about things that we are trying to do. Jump and run, use our skeletal muscles to get stuff done. It's also talking about things that you probably have to actively think about to control. When we talk about our involuntary movements, this is like our autonomic nervous system, so our cardiac muscle, which is, again, our heart beating, right? So that cardiac word, we've seen that before, right? That means our heart. Also our digestive muscles, our smooth muscles. There are various glands that need to move and release things in our body. So anything that you don't have to actively think about is also part of that motor system, just the involuntary section of it. All right, this next section, we're going to dive a little bit deeper, get a closer look at the anatomy of our nerves. So what's happening at the cellular level in our nervous system? Well, first of all, one nerve cell is called a neuron. A neuron can generate and conduct impulses right? So we're seeing this word again, right? Impulses or like signals would be another word there. They are communicators in the central nervous system and peripheral nervous system. They vary in size and shape, but they do always have some common features. And you can see that in this image off to the left of your screen here. They always have a cell body. This is what contains organelles like the mitochondria, things you've learned about in ninth grade bio or AP bio. We've got dendrites on every neuron, so you're going to see that often. That's what these little like branching things up here are, right? So that look like a tree branch branching out. We also are always going to have an axon, which is this long part of the cell here. We have those myelin sheaths, which are these yellow things you can see. Those myelin sheaths are like a nice protective coating for the cell, so that way our signals can move without any problems. And then when you see that there's like little tiny spaces in between those insulated sheaths or that protective coating, those are called the nodes of Ranvier. And all these parts are going to make up one nerve cell. So one singular nerve cell is going to have all of the things that we see here. So all those common features. The other part of the nervous system that we're going to look at is neuroglia. This is like nerve glue. They're little cells that are going to always help out our nerve cells, so they might not have a direct role in communicating like the rest of our nerve cells do, but they really keep our nervous system intact. There are 6 types of neuroglia that we are looking at. We are going to talk about microglia, oligodendrocytes, astrocytes, ependymal cells, Schwann cells, and satellite cells. And the main function of these is really to protect, insulate, and just have like general support. So if we're going to remember anything about neuroglial cells, they are here to provide just like a little bit of sidekick energy for our nervous system. The first one we're looking at, microglia. This is in the central nervous system only. It is small, it's like kind of shaped like a spider. We can see one right here in this image off to the side. And this develops from white blood cells, which work with our immune system. This cell removes bacteria, other cellular debris. So if a cell dies and parts of that cell are around, it's going to remove that so it doesn't accumulate and cause any like mess ups in our system. This is also what would form scars in any damaged areas in our nervous system. So my favorite, the little helper ones, make sure our nervous system doesn't have any bad bacteria around. Thank you. Thanks to those microglia. The next one we're looking at is oligodendrocytes. These are also only in the central nervous system. And I didn't point this out before, but you can see, right, that's why this little icon over here is showing that the brain and spinal cord are the only ones highlighted, right? Because that's where we're going to find these things. That's the only spot we're going to find them. We'll find these along the axon of neurons because they are producing the insulating layers of that myelin. And myelin's very important for keeping the speed of our impulses or conduction going in our neurons. So if you want to think about snapping your fingers and do it, that can happen so fast. If we didn't have that protective myelin coating, it would not work out as well. So a big thing here is it's going to like increase the speed in each of our nerve cells. We also have astrocytes that are also only located in the central nervous system. They are star-shaped. You can see them kind of connecting over here to like other nerve cells as well as our capillaries. There's a bunch of these in our nervous system. They are the most numerous, and they're going to prevent capillary leakage from happening. They also maintain our blood-brain barrier, which keeps our brain like safe from things. and help regulate what nutrients, what ions, what good things need to be in balance for our brain. All right, finally, for the glial cells in the central nervous system, we've got ependymal cells. These, again, only in the central nervous system. They form an epithelial-like membrane that's going to line cavities in the brain called ventricles. It's going to also line some cavities in our spinal cord. So you can see they like pack together really tightly. They produce cerebrospinal fluid, which is what keeps our brain floating nicely. It also like really helps prevent damage if we were to like get hit by something or fall over or have some sort of collision. So they are there producing that important fluid for us. The last two neuroglia cells that we're going to look at are Schwann cells. You can tell that we're in a different area of the body now because Schwann cells are only found in the peripheral nervous system, right, which is all these like nerves that branch off from the center of our body. So not in the brain, not in the spinal cord. This produces myelin sheaths in just the peripheral nerves. And what's different about this one is that peripheral nerves can regenerate, but the ones in our spinal cord and brain cannot. So when we think about like cells dividing or undergoing mitosis, right, these ones can divide still. So if we have some damage, these Schwann cells can help make myelin sheath in those peripheral nerves if they do need to regenerate. But what's not so great about neurons in our central nervous system is they cannot regenerate, which makes it dangerous if we damage anything in our brain or spinal cord. The last one is our satellite cells. They are also just in the peripheral nervous system. This is just creating like a protective coat around the cell. So you can see like we have our neuron cell right here, and those satellite cells are the purple ones like on the outside. It's just protection for our nerve cells in our peripheral nervous system. Next, we're going to talk about communication or how nerves send signals to each other. I think this is the most interesting, plus maybe also the most difficult concepts. So buckle up. All right. Communication we can talk about really in two ways in our nervous system, so that's how I've broken it down. The first way we're going to talk about this is action potential, which is just electrical charges inside a cell. I think this is a tough concept to grasp, so if you're somebody who feels a little lost, it's okay. We'll come back to it and keep talking about it. The second way we have cell signaling going on is with neurotransmitters, or just its chemical messages from cell to cell. So we're going to look at two types. The first type we'll look at is action potential. The second type we're going to look at is how neurotransmitters are sent from cell to cell. All right, this first one, action potential. So the big idea here is that this is just how a signal travels down one singular nerve cell. So we're only talking about one nerve cell when we talk about action potential. And really, each cell has the potential for charges to move across that cell membrane. So what we're seeing over here is that cell membrane. And what we're seeing inside the cell is that it's more negative, and outside the cell is more positive. So when we have a difference in charge like this, that can create an electrical current, and that current is really how our cells are going to send signals. Those charges come from different ions or different like particles that have either a positive or negative charge. So in the case for us, we are looking at potassium, which is represented with a K. We're looking at potassium ions and sodium ions, which is represented with an N, A, sodium. So if our cells can change or like let any of these ions go back and forth, that's how it can change its electrical charge. And that's really just the big idea. So now let's dive a little deeper. Outside of a cell, is positively charged. So we have more positive ions outside of the cell. And you can see that here, right? Here's the outside. And we see a lot of sodium ions on the outside. Most of the channels are closed, so sodium cannot get through those right now. When a cell gets a signal, when a nerve cell gets a signal, those channels open, which we can see here in the second one, right? And the sodium ions are going to diffuse into the cell. Once that happens, we've got more sodium on this side, so the cell becomes positive. As the cell becomes more positive, we're going to see sodium ions diffuse that way. There's also going to be more coming into the cell, and the cell's just going to keep getting positive all the way down. Finally, when those Na or those sodium channels close, we are going to see some potassium diffuse out of the cell. So on this last one, we can see our positive calcium going out this way to make the outside positive and the inside more negative again. And that cell is negatively charged again. So the changing of how this cell is charged is going to eventually send a signal all the way down that cell, from the very top of it to the bottom. And we'll see what that looks like. So once that signal gets all the way to the end of a nerve cell, now what? And this is where communication 2 comes in. So we've reached the end of the cell with that signal. Now what needs to happen is we need to pass that signal to the next neuron. And those neurons are not directly connected, right? So if we've got one here, and here's the end of it, the next nerve cell might be really close to it, but not completely. So that gap that you can see is called the synaptic cleft. We have to pass chemicals from one cell to the other. In this case, those cells or those chemicals are called neurotransmitters, and they're going to send a signal from neuron to neuron. So the end of axons have extensions or synaptic knobs with some synaptic vesicles at the end, and those synaptic vesicles are going to help us out in this exchange of information. And on the bottom here, you can see kind of like a big idea of what we want. We have a signal that's coming down the cell. We're going to pass it in this space here to the next cell so it can continue on. Pass it in this space here so it can continue on. Pass it in this space here. So when we are looking at the end of a neuron, that impulse has reached the synaptic knob at the end of that cell, we can see that there are some chemicals in those vesicles. Those chemicals are neurotransmitters. We can see them as those dots right here. And those molecules are going to get released into that space. So we can see that happening right here. All these little chemicals are getting released in this space. Once that happens, those molecules diffuse across that membrane where they can come attach to some receptors on the membrane of the next cell. So if they can attach, they are binding, right? So they are attached to that receptor, which is either going to stimulate an impulse or inhibit an impulse. So it's either going to keep passing on that signal, so it's going to pass on, or really like not have that signal happen or stop it. Neurotransmitters can be different chemicals, and those different chemicals bind in different ways, which is going to give us different behaviors and different communication. So one thing to be aware of here is that either we are going to get neurotransmitters that do one of two things. They are either going to excite the cell or make it more positive. So neurotransmitters can do this, or they're going to inhibit the cell and make it more negative. So the message might not be sent, or it will decrease the possibility for it to be sent. So neurotransmitters can do either this thing or they can do this thing, depending on what type they are. If you're somebody that's interested in like the type of neurotransmitter and what they do, this table kind of talks about what their actions would be on our nervous system. I'm not going to go through them all, but if you want to pause the lecture and read about them, it is kind of interesting. All right, our last portion of this, we're going to talk about drugs, or what different chemicals can do when they interact with our nervous system. The first one we're going to look at as an example from poison dart frogs, which is curare. It's a poison made from frog skin. It causes paralysis by blocking acetylcholine receptors at that neuromuscular junction, so between the nerve cells and the muscles. There's an indigenous group of people that have put that poison from the frogs on darts, which is how they get their name, and used in like hunting practices. And if you've been across the hall, you can see the non-poisonous poison dart frogs that Mr. Greshik has. Another drug that can affect our synapses and neurotransmitters is strychnine, which can be fatal to humans. It's often found in rat poison. If you inhale it, swallow it, absorb it through your eyes or mouth, it can cause your muscles to contract and contract and contract so much that you just stop breathing, which is all to do with how those signals are sent. So those signals are getting sent wrong in our nervous system. Finally, one that lots of people have heard of is the drug called alcohol. When alcohol enters the brain, it really does two things. It's going to bind with GABA receptors to make them more inhibitory, so those signals are not going to be sent with those receptors. And it's also going to bind to a glutamate receptor, so glutamate will not excite the cell. So it's going to really slow down those cell processes and those signals, which is not good if those signals involve like breathing or your heart rate, or things that we need to do to stay alive. So if somebody has taken too much alcohol into their system, that's something that can happen. If you're interested in learning more about this, one of our activities this unit will be looking at mouse party, so we can see the effect that different drugs have on the brains, specifically the synapses of mice, which can be similar to people. So get ready for a party. All right, that's all I've got. Thank you. If you've got your summary left to do, try to answer that essential question in your summary again. Otherwise, have a great day. Bye.